Let's focus today on the clinical interpretation of anti-drug antibodies. Let's begin with 7 statements. They are true or false. You can pause the video, you can take a look at them and try to answer them before we go on the talk. If you don't want to do that, just go ahead. I will be presenting the data that supports the responses, the accurate responses to each one of these as we go along on this talk. First of all, what are antibody responses? When we have a therapeutic protein that is perceived as an antigen or as a danger signal, the antigen presenting cell will digest it and present the T cell epitope to the T cell. And this is basically the way that the body is going to tell the immune system that there's something wrong with this drug, and it's perceiving the drug as a danger signal. So the T cell will mount an immunogenic response. The B cell can also be an antigen presenting cell. There's several different kinds of cells that present these epitopes to the T cell. But the T cell is central to actually mounting this immunogenic response. An immunogenic response is an unwanted, in other words, not desired, immune response to a drug. Once the T cell has been advised or warned that there is an epitope there that could be a danger signal and it's part of the drug, the T cell will react by producing cytokines that stimulate further that B cell, activate it, eventually makes it mature into a plasma cell, and this B cell response is characterized by the production of antidrug antibodies, which we often use the acronym ADA, antidrug antibodies. Furthermore, the T cells can maintain a level of cytokine production that will make that B cell mature into a memory B cell for a sustained antidrug antibody response. Now, let's not forget that the T cell response can also produce cytokines that will trigger what we call cellular immunogenic response, and the target cells that will now be activated to deal with the drug are neutrophils, eosinophils, macrophages. We can also activate mast cells, and this is in the absence of an antidrug antibody. The cellular immunogenic response is different from the B cell response. This talk will focus on the induction of antidrug antibodies and how the memory cells produce antidrug antibodies. How can we differentiate and interpret them? For further information on the cellular response to the drugs, the immunogenic response, and other aspects of the immunogenicity, please look at my other YouTube videos in my channel. I have created 2 playlists that could be important and relevant for your learning. And I will put all these links below in the description. Let's go over the primary antidrug antibody response. This is very similar to the primary antibody response to any other kind of antigen. For this primary antibody response to drugs, let's not talk about the memory cells right now. When the patient first receives the drug, and this drug is perceived as a danger signal, that naive B cell will identify, internalize, and present the appropriate epitope to the T cell. This triggers the activation of the B cells, which is what we saw in the previous cartoon. And this activation of the B cells eventually leads to the maturation into short-lived plasma cells in the lymphoid organs. This takes anywhere between 7 and 15 days after the drug has been given to the patient. That's when we see mainly the highest titers or the highest amount of antibody to the drug. And again, 7 to 15 days, and these are mainly IgM. In some cases, we could still identify some IgG antibodies. Over time, if the patient did not get any further injections, these levels go down as a result of long-lived plasma cells. I do want to mention that there is one case that this kind of timeline doesn't occur. There are some rare cases where the drug will elicit a type 1 hypersensitivity response, also called an anaphylactic response. And this is driven by IgE. And the timing is not 7 to 15 days. The timing could be 5 to 30 minutes. Let me just repeat that. 5 to 30 minutes. So if you want to know more about type 1 hypersensitivity response, which is also a type of antidrug antibody response and immunogenicity, please take a look at my other YouTube video. We're going to go on the more typical primary and secondary antibody responses, which are mainly IgM and IgG. Let's talk about secondary antibody response, and now we are going to talk about the role of memory B cells. So these memory B cells are the ones that are going to remain in the body, and on repeat exposure to the same drug, what happens with this memory B cell is that it will mature and produce the plasma cells that identify the same drug very quickly. First of all, the levels of the antidrug antibodies that are produced after secondary exposure are significantly higher than the levels on a primary response. We will talk about that a little later. But this cartoon really shows that there's at least a twofold higher if you see this. That, I mean, it's just a representation, because the y-axis could be a logarithmic axis. What we're trying to say here is it is higher. The levels are higher. They are now IgG, because this maturation has actually produced the switch to a more mature IgG. And also take a look at the time. This is the whole idea of having immunological memory to a potential danger signal. So this particular individual has identified the drug as a danger signal, unfortunately, and has mounted an immune response to that drug. So now it takes only anywhere between 3 and 5 days after the second exposure to have the highest titers. Eventually, these memory B cells will produce a low level of antibodies, but they're higher than after the first exposure. So if your patient is receiving the drug and it's having antidrug antibodies, these frequent injections will be producing higher and higher baseline levels of antidrug antibodies. This could eventually lead to many different consequences. The antidrug antibodies could be rendering the drug less efficacious, for instance. It may not be causing any safety issues, but eventually you could see that after years of treating a patient with a particular biologic, the patient may actually not be responding or losing the response, and you might need to consider another drug for that patient. So it is important to understand that on secondary antibody responses, the baseline levels that are driven by those memory cells start going up and up and up as the patient continues to have the stimulus of the drug that is identified by the memory B cells of that patient as immunogenic. Now let me just talk about timing. This is important because if you are designing a clinical trial and you happen to decide that you're going to take the blood samples for antidrug antibodies 15 days after the patient has been injected, and you're going to look at titers to define if there's a primary or secondary response, you want to know if there's a memory response to the drug. Again, you design your trial and you decided 15 days after injection. Look at what happened with your decision. You are actually getting the same titers after this primary and secondary response. You could erroneously be thinking that there's no memory and that the titers are not increasing. So you need to make sure that you tailor your clinical trials, understanding when is the best timing to obtain the clinical samples so that you could see a significant difference in titers to identify a potential memory cell. It is also very important that you always consider baseline levels before the drug is actually given. In the clinical setting for individuals, this is a little bit more tricky. So you need to tailor it case by case. But one thing that's important is that you obtain anti-drug antibodies of a patient, knowing A, why do you need them? B, what are you going to do with the results? And C, when are you going to be asking the lab to draw the blood for this patient? So all of these you need to take in account for clinical interpretation. So the clinical interpretation of antidrug antibody results needs to be done in context of the primary and secondary B-cell responses that we reviewed. What do I mean? Well, you're going to get antidrug antibody results and you may get an amount, a titer. So that you might be able to say, well, you have some parameters, they may give you the normal values, the values that are high, but do you know when was that sample taken with respect to the exposure of the drug? So those two things have to be taken together to understand how to interpret this titer. Also, you need to know baseline if available, especially if you are planning a clinical trial. Then you need to make sure that you consider the times that you are going to get those antidrug antibodies with respect of the clinical trial schedule of dosing for the patients. If you are interpreting clinical trial results retrospectively, a clinical trial that you did not design, or results that you are given for analysis, you do need to try to find out the timing of those antidrug antibody samples. Otherwise, the titers really are very hard to interpret. If you are getting just a positive or negative, and you don't know when those samples were taken, there's very little value and there's very little you can interpret clinically from what you are being given. So this is why my commentary on antidrug antibody results for clinical interpretation is that we do need to have the anchor of titers and timing. The other thing that we need to understand is if we are looking at different antidrug antibody assays. And this is where it can get a bit complicated because you might say, well, I don't know about the assays. I just want to see the result. Why would I have different assays? Well, the most simple response I can give you as an example, you could have an assay that's giving you IgM and IgG separately. So if you are looking at antidrug antibodies that are the IgM type and there's 0 IgG type, you might be able to assume, this is a primary response. So there might not be a memory response. If you get IgG, you are looking at most likely the antibody response, which is a mature response. Because remember that on the primary response, the levels of IgG are very low. It's majority IgM. But anyway, all of that you need to take into account. Now it gets more complicated when we talk about different antidrug antibody assays that are not by isotype. You need to know, is this a different assay? And this is where things get a little bit complicated for clinical interpretation, but we do need to make sure that we tap into the appropriate colleagues or experts that can help us interpret. Let me walk you through that difficult path. Was there a change of assay? When I get an antidrug antibody result, I normally never ask myself what assay they used. And perhaps even if I actually can see what assay was used, I may not have the ability to interpret what does it mean. Let me refer you to Boudreau et al. Publication in Clinical Experimental Immunology of 2018. He looked at several conditions of rheumatic diseases and patients that were treated with different drugs. For instance, in this case, we're going to talk about infliximab or adalimumab. And what he's presenting, his group is presenting on the y-axis is percent of antidrug antibody positive patients. And this is from many different publications. You can see there in a small text, it's a patient, the pooled patient population across the studies was almost 4,500 patients. And so what they did was to pull different papers and publications and say, well, what are these publications telling us about the percent of patients that mount antidrug antibodies to infliximab? The bubble size denotes the number of patients that were assessed for antidrug antibodies. So this is the denominator. And the y-axis is the numerator. What was the percent of those tested patients that were reported positive? Now the colors are different technologies or platforms that were used to assay for antidrug antibodies, and I don't know even half of them. So this is just to show you that there's many different ways in a laboratory to detect antidrug antibodies. And it changes over time. Not surprisingly, we are seeing more and more technologies as the time evolves because we get better in laboratory for assaying different things, and in this case, antidrug antibodies. Now, I just want to show 2013. What you can see there is a lot of variability. You can actually see that the percent of patients that are reported with antidrug antibodies goes anywhere, if my eyesight is not very bad, anywhere between maybe 10% to 60%. That's a big difference in terms of a clinical interpretation. Now, just take a look at all of those that are ECL, electrochemiluminescence. And you see that purple circle or bubble. Well, it's not that all the same assay gives the same result. That's not true. You can even see in 2013, I can see two bubbles that are ECL based, and they give very different results. When we look at the adalimumab story, of course, adalimumab was approved later, so the story begins later, 2002. But let's try to look at the same year, anywhere, 2013. Of course, his graph, that's where he's got it. So first of all, for anti-drug antibodies to adalimumab, I can see there's less platforms, less technologies than I can see for infliximab. But the variability, again, goes, in this case, from 0 to maybe 55%. I mean, in one of the cases in 2007, it seems that it really went, higher than 80%. So is this something that we need to be aware of? Yes, I think we are. If we are talking about clinical interpretation of antidrug antibodies, we need to understand that these are fields that are constantly evolving and the technology is getting better. So we need to understand how to interpret the results that we are seeing. If we are planning a clinical trial, or if we're planning a clinical program that's going to have 10 or 20 clinical trials for development of a particular drug, we should always pay attention to the technology or the assay or the platform that we're going to be using for antidrug antibodies. And if there is a change, we do need to make sure that we report it to the regulatory agencies when we submit the dossier for approval. If you are interpreting from the label or from publications because you want to treat a patient, you also have to take into account what exactly is this information telling you? What is the message? What is the real percentage of patients that create anti-drug antibodies? I don't know the answer, but it's very important that you realize that it's not a straightforward, simple interpretation. You need to understand all the elements around it. So talking about different assays, different anti-drug antibody assays, yes, we know there's assays that could be giving us different isotypes, and they're validated. When we get them, we can have, the anti-drug antibody IgG, anti-drug antibody IgM. Was there a change of assay? Yes, we could have validated ELISA results and later on validated ECL results, electrochemoluminescence. So we can have all of these in a different drug product or approved drug or clinical program. Even in one clinical trial, we can have different assays. In addition, we may have in a particular clinical trial or a particular drug, we can actually have anti-drug antibodies that are to different drugs. And these would generally be different anti-drug antibody assays, but they are validated. So what am I talking about? Let's just go over a primary response, let's say, to drug A. And so we have anti-drug antibodies to drug A, the patient is exposed and they get a primary response. That has, the typical elevation of titers or titers that you can measure. In this case, this particular cartoon from Benjamin Cummins, it does have some units in the y-axis. And is detected again anywhere between 7 and 14 days after exposure to drug A. If there's a second exposure to drug A with now combination of drug B at the same time, we could easily have a secondary response to the drug A where the titers significantly increase. Note that it is a logarithmic scale. And we now could develop also a primary immune response to drug B. And those would be reported separately, and most likely the same blood sample will be tested for the two of them, and we have a predominant titer in one that is a secondary response, let's say, to drug A, whereas drug B is already creating a primary response and eventually could have secondary responses to both drug A and drug B. So, where can this happen? Is this unusual? Well, we're seeing more and more drug combinations, especially in many oncology programs where they are biologic drugs, and oftentimes one is introduced before the other. So in these cases, the patient should be followed for the two different anti-drug antibody assays, just potentially to identify if a patient is mounting immune response to either one or the two of them in combination. There's also drugs that are more complex biologics, and many of these require polyethylene glycol, PEG, and these are called PEGylated drugs. And you could potentially have anti-PEG antibodies, and you could have them actually at baseline because there are many products that contain polyethylene glycol. And we could have pre-existing anti-PEG antibodies that could then identify a PEGylated drug as a potential antigen and develop now antibodies to the drug. As I mentioned, the B cell response is polyclonal, and there can be what we call epitope spreading. So if you have B cells, memory B cells to PEG, and now this PEG or polyethylene glycol is joined with a drug, with an active piece of a drug, our B cells could be spreading to the whole drug. So we also need to understand if there's pegylated drugs, we do need to have the detection of antidrug antibodies to both PEG and the drug, and sometimes even a third one to the pegylated drug as a whole. Complex biologics that are combining different pieces of different receptors could be, or one arm could be an arm of an antibody, like the FAB portion, and another one could be a receptor. And in these cases, we may also encounter anti-drug antibodies to the different elements of these complex biologics. So we may not have only one anti-drug antibody result, we may have different ones. And there's drug transitions with the entry in the market of a lot of biosimilar drugs. There's more and more transitioning from the biologic to the biosimilar. And who knows, maybe from biosimilar to biosimilar. So things could be happening that way, and an anti-drug antibody could actually identify one and not the other. So we may need to understand what anti-drug antibody we are measuring. And we need to understand if the assays are different, we do need to know if there was a transition from one drug to another. So in these cases, we do need to put it into context of what are we assessing and what kind of treatment is the patient receiving. So it is a complex, it's a complex interpretation, but it needs to have a lot of different elements to be able to dissect which is the message behind the data. And again, baseline levels are super, super critical and important. Without baseline levels, we cannot really interpret, because there's no, this is not like hemoglobin that we have a normal value for the whole population. These are different assays. These are anti-drug antibodies that normally should not be present. So we don't know how to put it in context if we don't have baseline anti-drug antibodies. So don't discard baseline levels and use only drug-induced. Baseline are critical. In some cases, like I said, in the clinical setting, we may not have baseline anti-drug antibodies, and in those cases, the best we can do is call the company that is doing the anti-drug antibodies for help with interpreting the results. Let's take a look at the first few questions that I had posed at the beginning. Number 1, primary anti-drug antibody response, IgM, can be detected 7 to 15 days after the first drug administration. That's true. A secondary memory response can be easily identified by a booster in titer anytime after the second exposure. This is false, because it's not any time. As I mentioned, if we take it after, let's say, 15 days, the titers could be very similar to titers after 15 days in primary response. So we do need to take into account that the secondary response is faster, and if we need to identify a booster in the titer, we need to make sure that we collect the sample sooner after that second exposure. Anti-drug antibody titers over time should be characterized during clinical development of biotherapeutic, potential immunogenic drugs. This is true. They should be characterized. And even though in old times, maybe the titers over time are not available to us, especially for some old biologics, more and more we should not just be happy by looking at percent patients with ADA positive for a given drug. That really means nothing. It should be percent patients at a given time. So after the first exposure, after the secondary exposure, how many days, and if they become negative, when. So the time frame is as critical as the titers for analyzing antidrug antibodies, and in more recent drug development programs, it should be fully characterized before approval of the drug. There should be only one validated antidrug antibody assay during drug development tailored to each biotherapeutic drug. Well, no, there's many validated assays, as I mentioned. First of all, we could have isotypes. So we could be using one platform and detecting IgG and IgM. So those could technically be considered two assays. Or we can have several platforms, or if there's a combination or a complex biologic, we may need to have actually different validated antidrug antibody assays during the drug development of any given drug. Antidrug antibodies. Let's understand what is being tested. So let's begin at the beginning, very basic. How do biologic drugs generally work? Well, we can have the drug, which can be a complex biologic, an antibody, or endogenous protein. So I'm just putting them in green. The letter Y denotes like an antibody that could be complex antibody or recombinant antibody, bispecific, or simple. And the other globular protein in there, or it could be complex biologic. Anyway, we have the drug. The drug has actually been tailored to bind a target. So what is the drug target? Well, the drug can bind soluble targets, or it could bind targets that are bound to a membrane. And depends on your drug, they will be binding sometimes only soluble, sometimes only bound to surface, or sometimes the drug target could be found both as a membrane bound and soluble. So we need to understand where those antibodies are going to be binding. Now, what is the antidrug antibody? Okay, so now we have the drug and we have the drug target. When there's antidrug antibodies, these antibodies could actually be binding the functional site of the drug. So whichever is the piece of the drug that bound the drug target. So if the antidrug antibody identifies that functional part of our drug, that means the drug cannot bind its target. So again, these are antidrug antibodies that would neutralize our drug. Now, not all antidrug antibodies are going to bind that particular piece of our drug. Some antidrug antibodies can actually bind a structural, not a functional piece of our drug. And in some of these cases, our drug can still bind the drug target. So not all antidrug antibodies will render the drug ineffective. Sometimes we may have circulating antidrug antibodies. They could even be high levels, and the drug can work perfectly well. So when you are assessing the results of antidrug antibodies, don't assume that there's going to be less efficacy just because there's antidrug antibodies. You do need to understand if these antidrug antibodies are causing an effect that's a negative effect of what you expect the drug to be doing both in efficacy and safety. So that's a second piece of interpretation. But let's take a look how antidrug antibodies are detected in general. I'm making this tape in March of 2021. I will give a brief explanation of the mesoscale discovery platform, how it works. We have in green a protein therapeutic that is captured on a solid phase. So in other words, we have the plates in the lab and they have been coated with the drug. We use blood from the patient. We wash these plates that are coated with drug, and if the patient has antidrug antibodies, they will bind to that drug that's on the plate. If the patient's serum sample doesn't have antidrug antibodies, they don't bind. Then we come back with drug that's been labeled, and if there were no antidrug antibodies bound to the plate, then that labeled drug will wash off and there's no reading. If, on the other hand, there were anti-drug antibodies stuck to the drug on the plate, then the labeled drug will bind and there's a reading. That's as simple as that. Simple, but not easy at all. Let me talk about just a couple of challenges. You will be hearing about target interference and drug tolerance when you get to analyze. You're given results of anti-drug antibodies to discuss and analyze. So you need to understand at least that there's something that you need to pay attention to. Target interference. The drug has been engineered to bind a target. If this target is a soluble target, it will be circulating in the blood of the patient. And of course, it will bind the drug. After all, the drug was designed to bind that target. So when the target binds the solid phase bound drug, it's available for the labeled drug to bind. So there's no anti-drug antibodies in this example, but we will have a positive result. So in target interference, we're talking that if there's a soluble target, there's going to be a false positive result. And this is where the validation tables that you will receive will tell you the conditions of the assay. Our colleagues in clinical assay and clinical analytics, they develop and validate these assays that are tailored to each one of the new drugs and each one of the targets. And there's different ways to get rid of that soluble target in the blood sample from patients without getting rid of anti-drug antibodies. So those are different protocols. You could use acids or incubation with soaps or different kinds of things that can get rid of the target or dilute it sufficiently so that they don't interfere, producing a false positive result. What is drug tolerance? Well, let's say that this particular individual or patient example that I'm putting here does have anti-drug antibodies, but this patient also has a lot of drug in the blood. So the anti-drug antibodies are actually bound completely or partially to the circulating drug that they were given. So when we come back with labeled drug, it doesn't bind because the anti-drug antibody is already occupied by the drug in the blood and maybe some of it bound to the plate, but there's no more available anti-drug antibody to be identified by the labeled drug. So drug tolerance is when there is drug in the blood that actually will produce a false negative result. So again, there's protocols that are tailored to each particular drug for each particular anti-drug antibody assay that can get rid or resolve the issue of drug tolerance by getting rid of the excess drug in the blood of the patients without destroying the anti-drug antibodies. And again, these are unique for each one of these assays. Let me just explain that about 10 years ago, or even within maybe the last 5 years, one of the ideas was to use 5 times the half-life of a drug before we take drug samples for anti-drug antibody testing. This would definitely make sure that the patient has either no drug in the blood anymore or very little. If we assume that we have a drug that has 5 days of a half-life and you wait 5 times 5 days, if you take the blood sample 25 days after the injection, you are way past the peak amount for either primary or secondary responses, and you may be missing the reading of good anti-drug antibody responses. So you do need to take into account not only the conditions of the assay and when you need to have very little drug in the blood and very little target, soluble target, but you also need to remember the kinetics of the primary and the secondary B-cell responses of human beings. So with all of that, you do need to understand if you are using an assay that allows you to take the sample soon enough after the injection of the drug, but there's a protocol that can get rid of drug in the blood when you're testing these antibodies so that you don't get false negatives in the assay. The assay for antidrug antibodies, when it's not a protein but an antibody, the drug is an antibody, is basically the same idea. So it's basically we have the drug, which is an antibody bound to the plate, the sample from the patient, and then the drug, which is an antibody labeled. And we have exactly the same challenges. If it's a soluble target, we could have a false positive result. And if there's a lot of drug in the blood, we could have a false negative result due to drug tolerance. And again, all of these assays are validated with pre-treatment of the serum samples that is tailored to each one of the drugs and each one of the targets that we're working on, so that we can have a better idea of true positive and true negative results. Now, how do we know if these anti-drug antibodies are actually neutralizing the ability of the drug to bind its target? Well, that's what we call neutralizing anti-drug antibody assays. So the anti-drug antibodies can be neutralizing, or they can be anti-drug antibodies that do not neutralize. How do we know if they're neutralizing? When we get positive anti-drug antibody results, as shown before, what we do is either our drug, either a protein or an antibody, is bound now to a solid base. We have the labeled target in this case, and we have a function. So this is what the binding of the drug to the target should be doing, and we can read this. The signal is actually measurable. Now, if we have an anti-drug antibody that neutralizes the drug, in other words, blocks the labeled target from binding the drug, then we don't have a signal. So the neutralizing antibody tests are complex. They could be on solid phase, or they could be cell-based. When we have a cell-based neutralizing assay, this means that the target of this drug is on the cell, bound to a cell membrane. And when we put the cells on the plate with our drug, there should be a signal that they're binding. If there's neutralizing antibodies in the blood of the patient, then this signal disappears. There's a lot of different neutralizing antibody assays. The thing that's important to remember is that neutralizing antibody are a subset of the anti-drug antibodies. In other words, some anti-drug antibodies may neutralize depending where they bind the drug. So the first thing that we normally do is detect anti-drug antibodies. If there's none detected, there's no need to do the neutralizing anti-drug antibody assays, because neutralizing anti-drug antibodies are only a subset of anti-drug antibodies. There are many different ways of doing these neutralizing antibody assays. It is even more complicated than anti-drug antibody assays. The neutralizing anti-drug antibody assays require to really tailor to the mechanism of action, to the actual target of the drug, and which kind of cells can be used, and then to the different kinds of ways that we have to detect that binding of the drug to the cell surface target. So there's many different technologies and platforms for the neutralizing antibodies, and you do need to understand that you're using the most up-to-date for your trials, validated, or if you're interpreting data from publications or trials that you did not design, make sure that you can interpret exactly which neutralizing antibody assay was used. Let's take a look at the last three questions. There are two types of antibodies to drugs. They can either be antidrug antibody or neutralizing antibodies. It's not true. The neutralizing antibodies are a subset of antidrug antibodies. Not all antidrug antibodies are neutralizing, but all neutralizing antibodies are ADAs. Drug tolerance means that when drug is present in the blood, the results will be false negative. Yeah, that's true. And if the explanation was a bit difficult, you can just rewind and go back to listening to the explanation. Detection of neutralizing antibodies is done with cell-based and non-cell-based assays, tailored to each drug program. Yes, this is true. So what do we need to know for the clinical interpretation of antidrug antibodies? Well, first of all, antidrug antibodies are antibodies. These antibodies are a result of a humoral immunogenic response to therapeutic drugs, in other words, B cell activation. So this does not cover any cellular immunogenicity that could happen in the absence of antibodies. Primary antidrug antibody response is different from secondary antidrug antibody response, which is the one that has shown adaptive immunological memory. This is important when we are looking at a particular drug. Is this going to create a permanent or persistent anti-drug antibody response? And then the next question is it causing any trouble? And that is in other video. The other thing that we need to remember is the anti-drug antibodies are antibodies to a drug, and this drug can be an endogenous non-antibody protein. And in this case, we need to be careful. There's a lot of issues about mounting an anti-drug antibody to an endogenous protein like insulin, for instance, because we could be generating antibodies to self protein, and that can cause very important follow-up safety issues. So just make sure that you understand what drug you are using when you're interpreting these anti-drug antibodies. Now, the drug can be a monoclonal antibody, and that's where things get tricky when we're looking at antibody to antibodies. It's a little bit like a house of mirrors. So just make sure that you remember which antibody you are looking at. Is it your drug or is it your antibody to the antibody? They can also be complex proteins, in which case we also need to understand what are the anti-drug antibodies, which piece of that drug are they binding. Thank you for watching. To create this video, I have consulted many different references. These are the ones that I would recommend, but I want you to pay attention to the last one in bold. I have created a YouTube video that explains the most recent FDA and EMA. So Food and Drug Administration is the regulatory agency that governs or regulates the approval of drugs in the United States and many countries. Many other countries use the FDA recommendations. And the European Medicines Agency is the regulatory agency that covers or regulates the drug approval in Europe, many countries in Europe. Now, they both have issued several immunogenicity guidelines. They go from 2012, 2014, 2017, and the most recent one, 2019. They don't substitute one another. They are complementary, and you need to be aware of all of them. So what I did with that video was to explain what is in these guidelines and what you need to take into account to bring the immunogenicity information of a new drug for successful approval. So if you're interested in knowing how this is done, you can watch that YouTube video. And if you are in the business of drug development and you are working with potentially immunogenic drugs, you need to take a look at those guidelines. Thank you very much, and I hope that you stay safe and healthy, and you can check other videos in my channel. Thank you.